Hello Expo judges, my name is Charles and this is my project on the DNA barcoding of juvenile fish. I did this project at Shoreline Community College and I decided to do it because I heard about their during school project time and I had already taken a camp during the summer, so I thought it'd be pretty fun. So when I went to Shoreline to do this project, I decided to barcode juvenile fish. And you might ask, why juvenile fish? Well, it's because Dr. Jan, my instructor at the college, had told us all about the Orca program and how they needed information. And, I mean, what a better way to use my research time than to help another organization figure out what they need out in the real world. So, the first step in DNA barcoding is extracting the DNA. And once you have successfully extracted the DNA from the extremely small juvenile fish, as seen in this picture, and this picture, you use a process called PCR to replicate the DNA. Once you have replicated the DNA, you use the nanodrop, which is a device used for measuring the amount of DNA that you replicated, and it gives you a ratio of DNA to water, which is normally a pretty small ratio, but enough to get sequence out of. So then, I cut the DNA using enzymes, and put the DNA in agarose gel. So after cutting and running it to see if that the DNA was successfully cut and barcoded, cut and separated, I sent the DNA to an out-of-state lab to get sequenced. Once they sequenced the DNA at the lab, I got it back, and I got the actual sequences of DNA listed all right here. And once you get that, you have to go through and edit out all of the ends, looking at the peaks of the graph and determining which peak corresponded to which letter of DNA. So once you get all those ends sorted out of the way or removed, you can put the sequence like this into BLAST and it'll come up with which DNA or which animal species it represents. And it kind of sucked because halfway through, after I decided even to DNA barcode the juvenile fish right here, my project kind of took a turn for the worst, and I couldn't find the juvenile fish for 3 weeks. I had to ask the Orca program how to find them and if there were any strategies, and they didn't get back to me for the 3 weeks that I had previously mentioned. So I decided to DNA barcode the feathers that they had collected from the digestive tracts of otters. And it wasn't quite as interesting to me, but I went with it, and again, it didn't work out so well because this bottom one is the same as this, and I didn't get anything from it because, the top one is the same as the bottom one right here, and didn't get anything from it because it's bacteria. So nothing came out of it, no bird came out of it. All that I could find was bacteria, which is not a bird. Bacteria you don't see flying around with their wings and feathers. So I learned from that in order to DNA barcode feathers, you first have to soak them in ethanol to cleanse them of bacteria that are still living inside them. And so after the 3 weeks, the Orca program sent me frozen juvenile fish in containers that I could see and find. So I de-thawed them and started the DNA barcoding process. In doing this, once I got to one of the last steps, I accidentally put double the amount of water in, diluting the DNA too much. And this led to the sequence you see here. And when I sent that in and then got it back, there was no match for what sequence of DNA it was, because it was too small and too jumbled up. And it kind of, again, sucked not getting any results on the project that I had started. But overall, this experience has really opened my eyes and shown me that biotech isn't really what I want to do with my life. And I'm glad that I learned this early before spending and investing too much time and money into biotech. And I'm grateful that I now know that biotech isn't for me. Thank you for watching and listening. Goodbye.